Okay. Good afternoon, everyone. Today, my topic is genetic variants can predict the outcome of brace treatment in patients with AIS. The authors have no conflict of interest. It is well recognized that bracing is the most effective conservative treatment for AIS. The purpose of the brace treatment is to control the curve angle. However, still, nearly 30% of patients will progress even after bracing. It is reported several factors were associated with curve progression and bracing failure, such as the poor compliance and the low level of skeletal maturity and large initial curve angle. In recent years, evidence suggesting that several genetic factors play important roles in the curve progression. Among different genetic factors, the SNP is an effective genetic marker for predicting clinical outcomes in various diseases. It is reported that several SNPs are associated with bracing treatment, such as ER-alpha, TPH1, NTF3, VDR, and VDBP genes. The two studies are from our center. However, all these SNPs are not from GWAS studies, only using the candidate gene association analysis. So the power of these studies are not very strong. The purpose of this study is to evaluate the outcome of bracing treatment for AIS and to further investigate the association between AIS progression-associated SNPs reported by GWAS study and the outcome of brace treatment. Inclusion criteria: female AIS patients aged from 10 to 15 years old. Risser grade was 0 to 1 degree. Initial curve angle 20 to 30 degrees, and have good bracing compliance. This is a brace treatment protocol. During the follow-up, if the curve angle more than 40 degrees or the progression more than 5 degrees, it is defined as brace failure. The SNP genotyping was determined using the Techman SNP genotyping assay. A total of 5 SNPs were selected from previous GWAS studies that were reported to be associated with curve progression and have successfully replicated in different populations. One study is from our center. Results: a total of 259 patients were enrolled in the study. Among them, 30% of patients had curve progression more than 5 degrees, and nearly 36% of patients in the failure group underwent surgery at last. So the overall surgical rate was 11%. For these 4 SNPs in SOX9, in TPX1, in CHD7, and MIR genes, there was no significant difference was observed between the success and the failure group for the genotype frequency or the allele frequency. And for this SNP, RS1073445 in BNC2 gene, there was a significantly higher proportion of genotype CC in the failure group. Also, the frequency of allele C was significantly higher in the failure group, with odds ratio was 1.59. Discussion: it's reported the curve severity, curve type, and Risser size are associated with brace outcome. So in this study, to minimize the impact of these factors, we restrict the inclusion criteria to curve angle of 20 to 3 degrees and Risser size 0 to 1 degree. So there was no significant difference of curve angle, curve type, and Risser size between the success and the failure group. The BNC2 gene was first reported to be associated with AIS by the Japanese study. They found the SNP could influence binding of transcription factor YY1 and further regulate the expression of BNC2. Our previous work also showed a significant correlation between the BNC2 expression level and the severity of the curve. Also, the influence of this variant of BNC2 on the musculoskeletal growth had been conferred by the animal model using the zebrafish. The BNC2 overexpression was correlated with the severity of body curvature, and the knockdown caused smaller body size. So in conclusion, 30% of patients received brace treatment had curve progression, and the SNP in BNC2 was significantly associated with brace treatment outcome. Thank you.